So we're here at the SLAS conference in San Diego. I've been joined by David Micha. David, firstly, thanks very much for joining us. Could you tell us a little bit about what you do for molecular devices?
Sure. I lead one of the sales teams in North America, mostly cover the cold places, which is great. But so we get to spend a lot of time inside working with the instrumentation. And the instrument I have here today is the SpectraMax i3 plate reader with Minimax cytometer, and our newest product, which is our ScanLater Western blot analysis cartridge. So one of the great things about this product is it's a base 3-mode plate reader that comes with absorbance, fluorescence intensity, and luminescence optics built in. But then it also has 4 cartridge positions that allow an end user to install without ever contacting us other than to buy the cartridge. So no engineer makes a visit. They can install additional optics. Example today is the ScanLater Western blot cartridge, which would go right in the front of the instrument. And it expands the i3 beyond just reading plates. What we can now do are read mini blots. And we do it using a europium-labeled secondary antibody. So we get wide dynamic range for protein detection on the blots. And really great part is that unlike standard ECL chemistry, where you have to read the blot pretty quickly after you've done the hybridization and development, we can actually read as long as 30 days out past the time that the blot is probed. So you have a lot of time to work with. If you want to go higher throughput, we actually have a stacker that can connect it, so you can load multiple blots and do as many as 20 or 30 in a run. It really increases sort of the possibilities when you're looking at Western blots and how you're going to process them in your lab. In addition to the ScanLater cartridge, we have cartridges for lots of popular assays, several time-resolve fluorescence assays, time-resolve FRET assays, even other proprietary technologies, luminescence being a common one. But the other piece of the system that makes it really intriguing to a lot of principal investigators is the Minimax cytometer option on the bottom. So the two parts are actually independent, and they can be joined together in about 5 minutes. But the cytometer adds essentially a 4x microscope objective and a CCD camera to the system. So in addition to reading a population of cells with the plate reader components, you can actually take some pictures. And you can correlate the pictures with the data that comes from the plate reader. So it makes the system highly flexible, capable of doing lots of different assays, and a really good fit for almost any basic research laboratory. So if you do have any questions for us, molecular devices can be found on the web at www.moleculardevices.com or 1-800-635-5577.